Inside a living cell, everything is in constant, chaotic motion. To study these building blocks, scientists must force that biological chaos into rigid physical order. Mapping the three-dimensional structures of proteins is the only way to see the machinery of life and fix it when disease strikes. But seeing these molecules at an atomic level requires bombarding them with X-rays, a process that only works if we can lock thousands of them into perfect, repeating crystals. Proteins naturally resist this. They evolve to wiggle, bend, and float in water, making them inherently difficult to pack into rigid, motionless structures. Francis Crick famously noted that biological laws are often just messy evolutionary generalizations. They lack the strict, neat mathematical formalism found in physics. Because we cannot rely on tidy formulas to predict these behaviors, scientists must actively intervene and force these chaotic machines to surrender to physical laws. A typical protein molecule is covered in floppy loops and dangling ends that flail around. These appendages act as bumpers. When researchers try to pack them into a crystal grid, the ends smack together, pushing the molecules apart and preventing a solid connection. Overcoming this requires more than just careful handling. Researchers must go down to the genetic level and rewrite the protein's DNA blueprint to remove these sources of chaos. Mapping the machines of life means actively redesigning them. This process begins on a computer. Software scans the DNA code to hunt for the specific amino acids that allow for all that excessive wiggling. Targeted design involves intentionally altering isolated sections of the molecule to change how it behaves. Surface entropy reduction addresses this flexibility problem directly. Imagine trying to pack a cardboard box full of wiggling octopuses. They won't sit flush until you clip away their tentacles, leaving behind solid, compact shapes that stack like bricks. This surface entropy graph shows the molecular equivalent of that process. Scientists identify the long, flexible amino acids, represented here by these green clusters, specifically lysine, glutamate, and glutamine, and mutate them into short, rigid residues, like alanine. Removing these hyperactive appendages forces the protein to lose its flexibility, allowing it to finally slot into a dense, repeating crystal lattice. Some proteins are so complex or poorly understood that researchers cannot guess which parts to cut. When targeted logic fails, scientists use combinatorial design, a shotgun approach that uses accelerated, random evolution to find a solution. The primary tool here is error-prone PCR. By manipulating the chemical environment, researchers force DNA to make random copying mistakes, creating thousands of unique genetic variations. Scientists then screen these thousands of mutants simultaneously to see if even one of those random errors happens to produce a protein that can form a crystal. This can be pushed even further by shredding the DNA of the survivors and shuffling the pieces back together. This gene exchange creates new hybrids that combine the most beneficial traits of the previous generation. Brute forcing physical order often requires harnessing the chaos that created biology in the first place. In the laboratory, the first attempt at crystallizing a protein almost never works. This workflow shows the path to the final crystallization trials. Most projects hit a dead end at this green box. These red arrows represent the iterative cycle required to negotiate with nature, looping back to the beginning to tweak the DNA and try again. Eventually, the right genetic modification forces the protein to submit. The unpredictable molecule snaps into alignment with neighbors, forming a rigid, perfectly repeating grid. This crystal is then hit with X-rays, finally revealing the three-dimensional machinery that was previously invisible. The end result is the clean, colorful ribbon model used in textbooks and research papers. These models are more than just snapshots of nature. They are artifacts of human engineering. They represent the moment we outsmarted billions of years of evolution to force biological chaos into a form we can finally understand. To cure diseases or design highly specific drugs, scientists must first figure out exactly what they are targeting. They have to map the invisible, three-dimensional structures of human proteins. But normal cameras cannot capture this. Proteins are too small and vibrate too erratically. To read their structure, scientists must force these molecules into perfectly ordered, identical crystal grids, allowing us to trace their atomic outlines, like the blue electron density mesh mapped here. Without these crystalline maps, drug discovery relies on broad guesses rather than precise molecular engineering. Crystals offer the exact coordinates we need. But before you can grow a crystal, you need material. Acquiring enough pure, identical protein to build that rigid lattice is a massive biological hurdle. Historically, researchers tried to extract proteins directly from natural tissues or plants. Because target molecules are incredibly sparse in the wild, scientists had to grind through literal mountains of physical material, like these jack beans, just to scrape together a few micrograms. And the moment you break open a cell to harvest it, you release endogenous proteases. These act like biological scissors, instantly chewing up the target protein before it can be safely isolated. To get around this, structural biology relies on heterologous overexpression. Researchers take the DNA of the target protein, insert it into a foreign host cell, and hijack that cell into becoming a dedicated manufacturing plant. But biological hosts resist being hijacked. Flooded with unfamiliar blueprints, the host cell often panics. Its folding machinery gets overwhelmed, misfolding the foreign proteins and dumping them into dense, useless clumps called inclusion bodies. Simple harvesting yields almost nothing.
To get pristine material, scientists have to actively hack and debug the living factory. The default hardware for this process is the bacteria E. coli. It is cheap, grows incredibly fast, and handles genetic manipulation well. But E. coli is simple. When you task a bacterial cell with building complex human proteins that require intricate folding or specific post-translational modifications, the assembly line breaks down. To match the complexity of the target, researchers upgrade the host. They deploy yeast, which functions as a robust secretion factory, or mammalian cell lines that offer supreme versatility for processing human biology. When traditional cell vats fail entirely, scientists utilize alternative hosts, engineering specific proteins to be expressed in the milk of livestock, like these goats. Upgrading the biological hardware solves structural limits, but it cannot fix software conflicts hard-coded into a species' DNA. The genetic code might be universal, but dialect matters. Human DNA relies on specific nucleotide triplets, codons, that bacterial factories rarely use. When the bacterial ribosomes hit these rare codons, they lack the right biological tools to read them. These genetic software bugs stall the assembly line, bringing production to a halt. To clear the error, scientists use custom-engineered Rosetta bacterial strains. These hosts come preloaded with a patch of the missing translation software, allowing them to read human code fluently. And then there is the toxicity problem. If the protein you want to build is lethal to the host's internal chemistry, the factory will die before it manufactures your product. This graph illustrates a workaround called autoinduction. First, scientists feed the host glucose. The bacteria prioritize this rich fuel for population growth, ignoring the toxic blueprint. But once they exhaust the glucose, they switch to lactose. This automatically triggers a new set of genes, and production of the target protein suddenly skyrockets. By weaponizing the host's own metabolic instincts, scientists force the factory to build the very thing that destroys it. Even a successfully manufactured protein will often refuse to crystallize. Molecules frequently contain floppy, disordered ends that prevent them from locking into a neat grid. Through limited proteolysis, researchers use chemical scissors to strictly snip away flexible tails, leaving behind a rigid, stackable brick. Another major physical barrier is glycosylation. As we see in this model, eukaryotic proteins often emerge coated in messy, uneven sugar branches. These bulky sugars act like bumpers, keeping the proteins from packing tightly together. Researchers apply specific enzymes to shave these sugars off. A successfully crystallized molecule is rarely in its natural state. It is a heavily sculpted, hyper-rigid artifact, engineered purely for perfect geometry. Despite all these genetic patches and chemical trims, relying on living cells means constantly fighting against their unpredictable need to stay alive. To bypass the cell entirely, researchers use cell-free systems. They rip biological translation machinery out of the organism, letting it operate in a fluid void. Removing the cell membrane allows scientists to calibrate the environment directly. They can precisely add synthetic lipids, folding chaperones, and redox buffers to orchestrate assembly in a test tube. The result of this isolated expression is pure, concentrated material, clearly visible in these test tubes of highly expressed green fluorescent protein. Mapping the architecture of life is not achieved through lenses. It is the result of re-engineering biological machinery.